Hey guys, do you know that you are mostly a microbe? There are more microbial cells in your body than a cell. Microbes are found everywhere in our body, in streams, on rocks, on our mobile screens, even in our food. Despite their bad reputation, microbes are mostly beneficial or have a neutral effect on our lives. Hence, microbiology is the study of microorganisms which are too small to be able to be seen by a naked eye and include things like bacteria, fungi, and viruses. Accordingly, to observe this unseen world around us, I've structured an experiment which may clear some of our doubts. This experiment revolves mainly around three major aims involving many microbial techniques like streaking, sterilizing, etc. Firstly, we culture bacteria and yeast on a nutrient media. Here, we are using potato as a nutrient media. Secondly, we check the susceptibility of antibiotics on bacteria and bacterial resistance towards antibiotics. Lastly, we check the effect of disinfectants on most accessible places in our homes that may contain microbes. So in this experiment, we will require boiled potatoes, plastic bowls, syringe, yogurt stock solution, yeast stock solution, and many more basic microbial equipments. In the first part, we boil the potatoes and cut them into slices, setting them aside to cool. Next, we streak the bacterial and yeast stock solutions on the potato and store them, sealing it up for the next 24 hours. We also keep a potato control for proving a hypothesis, which we'll observe after 24 hours. In the second part, we test the antibiotic susceptibility to bacteria, leading to which we follow the same protocol considered in the first part. And in addition to that, we just scoop out a well from the middle of the potato to check the zone of inhibition. We then add solutions in the well measurably. In total, we make 4 bowls, namely curd control, in which we add 0.5 ml warm water, antibiotic control, which consists of 0.5 ml antibiotic stock solution with no curd, and other 2 bowls with different concentration of antibiotic stock solution, 0.2 mg and 0.5 mg. Further, we seal them up and keep them aside for 24 hours. Lastly, we check the effect of disinfectants on these bacteria, proceeding to which we need to collect samples from the most accessible places in our homes, for example, remote, doorknobs, etc. After collecting the samples, we streak them on the potato and seal them. Then we spray the disinfectants on our object, again taking the samples and streaking them on the potato. Then we clean our working area as we don't want to encounter with any infection, and wait for the next 24 hours for the results to show up. After 24 hours, we observe that some white spots starts to appear on the potato, giving us an indication of the growth of bacteria and yeast colonies. Although we cannot differentiate between them naked-eyed, but yeah, we can see a color difference in both of them. We also see a white sheet on the yeast culture representing colonies. Proceeding in the next part, we observe a zone of inhibition on both the test potatoes, which was an obvious guess. Here, we spot a small zone of inhibition in the less concentrated sample and comparatively larger radius in the latter. But we see no inhibition in the antibiotic control and the curd control, proving that our hypothesis was right. And lastly, we can see the effect of disinfectants on bacteria, as we can easily make out that there are less number of bacteria grown on the disinfected samples than the normal samples. After these experiments are directed and observations are noted down, we have to discard these samples by dissolving them in bleach for 30 minutes and then depositing them. We do this in order by following our biosafety norms so that no infection is spread. Finally, while concluding the experiment, I would say that we have microbes all around us. May some are good, some bad, transient. Adding to it, antibiotics kill specific bacteria according to their concentration and dosages. And also, disinfectants and antimicrobial agents works to some extent to sterilize us and our surroundings. Thank you.